Hello friends, in this video lecture we will be talking about genetic code. What is genetic code is all about. We know that genes are the segment of DNA that produces a functional RNA. And the functional RNA, they include in general three broad categories of RNAs, the mRNA, the rRNA and the tRNA. Of these three, there are other smaller categories of RNA, of course, like the sgRNA, snRNA, snORNA, and the tmRNA and siRNA, miRNA. Different types of RNAs are there, but these three categories of RNAs are directly involved in protein synthesis. And the mRNA is the messenger RNA. It carries the message that is hidden in the DNA, in the gene, and it transfers the message to the protein. And this concept of transferring the message from DNA to RNA to protein has been called central dogma. The concept was given by Crick, and that the information is carried by the genetic information, of course, is carried by the DNA, and that is transferred to the mRNA via the process of transcription, this one. And the mRNA then transfers the information to protein. Proteins are made of amino acids like this. So each of the amino acid must have its counterpart in the mRNA, and each of the sequences in the mRNA must have its counterpart in the DNA. Then only it is possible to transfer the information from DNA to RNA to protein. So when it was seen that the proteins are made of amino acids, it was later on found that each amino acid is in fact coded by a 3 nucleotide sequence in the gene. Each of the 3 nucleotide sequences in the gene, they are responsible for coding for a particular amino acid in the corresponding protein, of course. These 3 nucleotide sequences have been referred to as genetic code, and they are also called codons, of course. So what are genetic code? Genetic code are basically nucleotide sequences in groups of 3. In groups of 3, because each of the 3 they code for, they actually bring three anticodon counterpart which are present in the tRNA, that actually bring the amino acids to be covalently tagged with one another, forming the protein. So because the tRNA has an anticodon comprising of 3 nucleotides, the codon also comprises of 3 nucleotides, and each 3 nucleotide is therefore responsible for coding for a particular amino acid in a protein. So that is called a genetic code. So the genetic code is triplet in nature. That means consisting of 3 nucleotides. What are the evidences that genetic code is triplet in nature? The first evidence is mathematical. DNA comprises of 4 nucleotides. RNA also comprises of 4 nucleotides, only T being replaced by U in RNA. Rest of the nucleotides are similar. But proteins comprise of 20 amino acids. So it is not possible for each of these nucleotides to code for 20 amino acids. Even if they are assembled in groups of 2, then also it is not possible for synthesizing 20 different amino acids. In groups of 2, according to permutation and combination, 4 to the power 2 means only 16 sequences are possible by all these 4 nucleotides. So this is also insufficient for coding for 20 amino acids. So the nucleotide codons must be present in groups of 3 for coding for all these 20 amino acids. In fact, they are more in number The genetic codes are more in number in comparison to the amino acids, the code. And it was later found that, in fact, some of the amino acids are coded by more than one codons. That is being called codon degeneracy. That we'll see later on. The next proof for this is Frameshift mutation. What is this Frameshift? What is frame? Frame or open reading frame. An open reading frame is basically a gene. It starts with an ATG sequence and it ends with an TAA, TAG or TGA. In fact, this T is U in case of RNA. That's why initially I called UAA, UAG or UGA. These are three stop codons. So an ORF comprises of a start codon, and after a certain distance of about 150 nucleotides, if there is a stop codon in any of one of them, then this will comprise an ORF. So in a DNA sequence, there may be different reading frames. So this reading frame, this one starting from the ATG, this is the correct reading frame. This will end somewhere after 150 nucleotides or base pairs when a TAA or TAG or TGA sequence arrives. But these reading frames, this one and this one, they might have started somewhere, maybe regarding this one, it has started somewhere with ATG. This has also started somewhere with ATG, and it will end somewhere else. So there are 3 reading frames for a particular strand, and for double strand there are 6 reading frames possible, and but only one of them will actually be coding for a protein. So a reading frame starts from an ATG and ends in a stop codon, one can say. A reading frame will change if you have an insertion or a deletion kind of mutation. It will cause a shift in the reading ORF or reading frame. But it has been seen after 3 nucleotides of consequent insertion or deletion, the reading frame is restored once again. The only difference is one amino acid will be extra. If it is insertion, one amino acid will extra be coded in the protein, and if it is deletion, one amino acid will be less in the protein. So that means if 3 nucleotides are added or deleted, the reading frame is restored. That also means genetic code comprises of 3 nucleotides. That is, it is triplet in nature once again. This is disturbing. The telephone was disturbing. Anyway, somebody is sending messages. I will see it later on. Now, let us see how it is happening. So this is a sequence, is a reading frame starting from ATG, CGT, ACG, TCG, etc. So the reading frame starts from here and goes onwards like this. It's a normal one. So an insertion has occurred here, due to which you can see the reading frame had changed from this place onwards. It is not now CGT, ACG. It is now ACG, TAC, like that. One nucleotide, one base pair, it has shifted forward. Similarly, when you add one more nucleotide is inserted, the frame has again shifted one nucleotide forward, but it is not the same like the original one. When another nucleotide is inserted in the place, that is consequently 3 nucleotides are inserted, from here the reading frame is restored. Okay, you can see it in the original sequence where ATG, CGT, ATG, CGT, ACG, ACG, TCG, TCG, TA, and TA afterward, whatever sequence is present, they will be coded. So you can see the reading frame is restored when these 3 nucleotides are inserted. Similarly, if 3 nucleotides are deleted, again the reading frame will be restored. But this will encode for another extra amino acid, that will be lysine, I think. That will be incorporated extra in the protein, but the reading frame is restored. That also means that this new nucleotide, the three nucleotides, they incorporate a new amino acid. That also means that genetic code is triplet in nature, and restoration of the reading frame is also a proof that the genetic code comprises of three nucleotides. The last proof is this triplet binding assay. Triplet binding assay. What does that mean? When it was possible to synthesize three nucleotides, synthesize mRNA in the laboratory by corona, in fact, then it was possible to synthesize mRNAs comprising of three nucleotides only. So all possible sequences, 64 types of sequences could be synthesized. Here I have given one example with AUG. And this is a binding assay on the membrane. This is a filter membrane, nitrocellulose or nylon membrane. And this assay is done many times. If there is this AUG sequence, this triplet serves as a codon, then only this tRNA, which is complementary to AUG. AUG codes for methionine. This will bind with this, with the codon. The anticodon of the tRNA will bind with the codon of AUG, and the ribosome will assemble for protein synthesis. And this will make a complex, and so it will not be filtered out. Since this tRNA is radio-labeled, the radioactivity will be detected in the membrane, proving that a complex has been formed. That means AUG codes for methionine. This is also proved at the same time. But if you add a different tRNA, that is not the met tRNA met, the amino acyl tRNA is a different one, not matching with the AUG, they will not assemble, and so they will be filtered out, proving that this AUG, or if you add here the code for alanine, the genetic code for alanine, then assembly will occur and you will find radioactivity in the membrane. Otherwise, any other codon which is not complementary to the amino acyl tRNA will not form a complex here, and so it will be filtered out. So this finally proved that genetic code is triplet in nature. Not only that, it was possible to prove by this experiment which codon codes for which amino acid. It was actually the last experiment by which the complete deciphering of genetic code or codon dictionary was possible. So now let us see, in our next video lecture we will see what are the salient features of genetic code. Thank you very much for your kind listening.